everyone for coming out. Great to see a good attendance. Always makes Josh more nervous, which is always good for a PhD defense. So Josh, this is sort of a strange experience because a number of us were here just a few weeks ago, and it's not normal, as an academic to have two PhD students finishing in the same month. But it's good. It's good. Don't worry, Kadar. And so back here again, second student. Each one's different. Each one has a different story. And while Kadar came to Utah to work with me, it was very much worked out beforehand. Josh was a different story. Josh made the mistake of thinking that biochemistry was interesting and that he would have fun in genetics and running biochemical assays and things. And it took a little while to see the folly of that approach. And so he arrived at my door, kind of unannounced. He sort of heard that there was stuff going on down here. He kind of arrived at my door, knocked on the door, and said, "Can I talk to you?" And like all puppies who kind of arrive at your door, you create a very special relationship with them. And even though you never set out to have a dog, even all of a sudden you have this amazing animal and partner and friend for life kind of thing. And so it's been with Josh. So it was never quite planned, never strategic on anyone's part, but it just worked out really well. And you'll get a little sample today of the breadth of what Josh has accomplished over the years here. I think at the end he'll give you at least a summary of all the scientific productivity he's contributed to. And that's really what's noteworthy as kind of a backdrop to all of this. Josh has just sort of gone from one project to another, and not just as a dilettante, but really made major contributions. He started with animal experiments, learned how to do all the tricks required with that, and I thought, okay, that's great. Josh has found his home. He's going to do animal experiments. We got a program. We're going to make this work. And then next time I turn around, well, he's actually working on humans now because he wants to do some translational research, and so he's taking images from humans and trying to figure that out. And I figured, okay, good, we got that fixed. Now Josh is going to do human research, and that'll be it. And then he said, well, I actually want to do computer simulations. And then so we started doing computer simulations. And so he's been kind of a moving target. It's taken a while for him to land, take ownership, and get some projects that are really his, that he owns to completion. But he's done that. But along the way, I think all of us who've interacted with him know to appreciate just how much he gives to research, how much he brings to the table. He's enthusiastic, he's diverse, he's willing to try anything, do anything, get up at any hour in the morning and stay up to any hour of the night to get the work done. And that's enormously valuable. And I think all of us who've worked with him have learned to appreciate Josh for that. So you get hopefully a good sense of the projects that he's really grabbed as his own and has ownership of, but against this backdrop of a productivity that's really remarkable for a student in this relatively short period of time. So with that, we'll let Josh take the floor after one more announcement, just to make sure everybody's clear on the procedures here. Not everybody's been to one of these or participated in one of these before. So we begin with an open presentation. Josh will present his research. The normal expectation is that if you have a question that's really important, like he says something you just don't understand and you're not going to get any further, you're going to be lost instantly if you don't get it, then feel free to interrupt. But we want to let him more or less do the presentation to the end. He's targeted, roughly 45 minutes for that. Then we'll have an open question period, which the audience is encouraged to ask questions. In fact, you may not get any more food unless you ask a question. And then we'll ask you all to leave, and then we'll have a closed session with the actual committee. And the committee members are all here in the front row. And Frank Sachs has been kind enough from Germany to stay up until, do the math, midnight, to start this defense. So he's on the line listening in, and he'll participate as we go along as well. And hopefully they'll stay awake. I think the presentation should keep them awake, right?
Try and keep it moving.
Okay, well, Frank, that'll be our mission today. And so with that, let's go ahead and get started. Thank you.
Okay, just as on that point, Frank, can we just make sure that you're hearing us?
I hear you very well.
Wonderful, wonderful. Okay, good. Just want to make sure we have everyone that we need. Okay. Well, Rob, thank you very much. I appreciate that introduction. And thank you to all of you for coming here later in the afternoon and spending this time with me. Today I'm going to be talking about enhanced interrogation of the cardiac electroanatomical substrate. Say that 3 times fast. But before we talk about the cardiac electroanatomical substrate, let alone start interrogating it, I want to focus on one of these words just for a moment. And this is the word substrate. What do I mean by a substrate? Well, I've got a story that can maybe help us understand this a little bit better. So last year about this time, a friend approached me about going on a hike with him. And I like hiking, so I entertained the idea. But his idea of a hike was to go from the north rim of the south rim of the Grand Canyon in a single day. Now, it'd been a long time since I'd been to the Grand Canyon, but my recollection of it was something on the order of magnitude of grand. And so this idea of having to go from one side to the other side of the Grand Canyon in the course of a single day was a little bit daunting, but interesting. So I mapped it. I went to Google and I found some information. I wanted to figure out what sort of effort might be required to make this crossing. I found out as a crow flies, it's about 12 miles. Okay, but we all know that hikes, any self-respecting hike, doesn't follow the path of the crow. So if we actually plot that, the actual course of this hike, we see it's about 22.5 miles. Now, from an engineer's perspective, those lines look about the same. Somehow, we've got another, there's another 10.5 miles somewhere compressed into this almost parallel path. I think this map is maybe missing something of interest with regards to this hike. So if we try another view, we'll see if we can get a better map. So here we have satellite imagery where we can see the north rim and we see the south rim. And okay, obviously Grand Canyon, we know that there's going to be some very interesting terrain, and this map does nothing but confirm that. However, in terms of visualizations or conveying information, the information encoded in this 2D projection of the Grand Canyon fails to really give us the critical information about what sort of effort is going to be required to do this crossing. So maybe there's a better map that we can try. And of course there is. Here we have what's called a topographical map. I'm sure you're all familiar with this. But in this topographical map, if we take a closer look, not only do we have the distance information encoded, but we can see these isoclines, where we have the altitude indicated on this map. So now we can really start seeing the relevant features of this hike that are of interest for our purposes. This, I would argue, is the substrate. This is the substrate of that hike. Okay. So what points I want to make here, one is about maps. Maps are very useful for conveying this type of information, and they'll come into play later. Additionally, a substrate is ultimately what makes something like this interesting. And when we have this information, we more often than not will have a successful result. And last year in October, we did have a fortunate result where the three of us started out with 3 and we finished with 3, made the crossing. And it was a great experience. But what does this have to do with electroanatomical map, or the electrophysiology of the heart? Well, it turns out in electrophysiology, there's what I wanted to refer to as the AF canyon. Now, over on this side of the AF canyon, there are patients with this arrhythmia called atrial fibrillation. Atrial fibrillation is the most common cardiac arrhythmia, leads to a 5-fold increase in the risk of stroke, and can initiate heart failure, and ultimately leads to a loss of quality of life for many patients. On the other side of this canyon is sinus rhythm. This is normal cardiac conduction. Hopefully everyone in this room is in sinus rhythm, and will happily stay there for a while. Now, the trick of this AF canyon is patients with AF are not able to make this transition on their own, but they need guides. They need people like this. Unfortunately, here at the University of Utah, we have some of these experienced and very well qualified cardiac electrophysiologists who help patients navigate this substrate to return to sinus rhythm. The challenge that these guides face is that no one patient has the same substrate. The relevant features necessary to navigate this crossing change from patient to patient. So as an engineer, what I want to do is I want to create maps of the electroanatomical substrate of the heart that can provide better guidance for these cardiac electrophysiologists to help patients in their transition from AF to sinus rhythm. Okay. Oh yeah, there are proper guides. So let's jump into what we're going to do here today. So we've already talked about maps of the Grand Canyon and talked about what substrates are. And from there, we moved into a little bit of discussion of atrial fibrillation. I want to provide a little bit more information about atrial fibrillation and arrhythmogenesis before we move into how we might generate electrical maps of the heart. Finally, we have these 3 aims where we talk about different techniques that may exist but could be refined, or that would be novel in the clinical setting in order to map the substrate of the heart. And as we end this discussion, hopefully we can compare and contrast these approaches and talk a little bit about if these maps are providing the relevant information that electrophysiologists need to help make this crossing. Okay, let's jump straight in. Atrial fibrillation, as the name implies, is an arrhythmia that is centered in the atria of the heart. So you have the ventricles, down here the primary pumps, and then the atria that reside above those. In normal sinus rhythm, the heart maintains a coordinated contraction. So the atria and the ventricles contract in a coordinated manner that is governed by the electrical conduction moving through the heart. In atrial fibrillation, that conduction is disrupted and the mechanical function suffers, and the heart races and all those other things that I mentioned before can begin to occur. So how does this even start? Atrial fibrillation is primarily initiated by mechanisms within the pulmonary veins. Specifically, there's electrical activity, I would say rogue electrical activity called ectopic beats that originate in the pulmonary veins that initiate this atrial fibrillation. This was first shown in work by Hasegaer et al., where they actually were able to record the locations of these ectopic beats and found that the vast majority of them occurred in the pulmonary veins. However, triggers are a critical component of arrhythmogenesis in atrial fibrillation, but there's another side to the story, and that's, of course, the substrate. Triggers alone, someone in this room during this talk is probably going to have an ectopic beat in their heart, and they're going to be just fine, we hope. But if someone has a diseased cardiac substrate like this, in this case, I'm showing you a visualization derived from an MRI in which we're showing remodeling of the tissue. It could be like fibrosis or changes in the spacing between cells or the connectivity between cells that affect the way that wave fronts move through that tissue. Now, when you get in the interaction of these triggers and the substrate, then you have the pieces in place to develop atrial fibrillation. So any given patient finds themselves somewhere on this scale. Maybe they're more trigger-mediated atrial fibrillation or more substrate-mediated. It turns out that patients who are more on this trigger end of the spectrum, they have a good prognosis. There's a treatment strategy called pulmonary vein isolation, where lines of scar can be created around the pulmonary veins that prevent the ectopic beats from propagating out into the atria and initiating atrial fibrillation. So this works pretty good with people on this end of the spectrum. However, those with a more substrate-dependent atrial fibrillation do not respond as well to this type of therapy, and new techniques are needed in order to guide treatment strategies for these patients. So again, what we want to do is we want to develop maps of this substrate that can provide guidance for treatment. Okay, so if it isn't clear, our focus here today is the substrate end of this spectrum. One more thing, though, on triggers and substrate, just to illustrate how they interact to produce an arrhythmia. Here we have just a real simple model of some scar tissue embedded in healthy myocardium. This could be from myocardial infarction or some other disease that has created this non-electrically excitable tissue. If we excite this tissue through normal mechanisms or sinus activation, we get a wave that moves through the tissue. This is the wave of excitation, that orange bar, and behind that is what we call the refractory period. Briefly, that orange bar is going to move through this tissue at a conduction velocity that's based on the connectivity of the cells and the underlying substrate, such that in a typical case, it might be moving at about 1 meter per second. However, in pathological cases, this can definitely be altered. The refractory period I'm showing here is a function of the excitation of the individual myocytes in the heart. So this is what's known as an action potential. And what happens here is cells that are at rest are negatively charged and experience a reversal of potential. And this reversal of potential is passed on to downstream myocytes. and is what causes this wavefront to move through the tissue. However, unlike muscle, skeletal muscle, this action potential is prolonged in cardiac tissue. And this generates this refractory period because until this myocyte returns to rest, it is no longer to initiate another action potential. So you kind of have this waiting period before it can fire another action potential. And we'll see why that becomes critical in just one moment. So we allow our conduction to continue to propagate through this substrate. You can see it's not exciting this tissue. Oh, but we have captured this channel through the scar. And maybe it's moving at a different speed. Maybe it's, well, it could be different, but not unlike this tissue, it's moving through that channel. And that's not a big problem in this type of conduction. Presumably, when that excitation tries to exit out of the scar, it's going to meet up with refractory tissue, and this red bar indicates block. Essentially, this tissue is refractory and is no longer excitable, so when that wavefront moves into that region, the excitation wave simply annihilates. Okay, but obviously arrhythmogenesis does occur, and here's where triggered activity comes into play. So we get an ectopic beat from some other location that sends another wave propagating into this medium. You can see it moves into this resting tissue just fine, and it blocks down here at this refractory border. If that wave then captures this channel, you have an open path for the reentry to start circulating around that arrhythmic substrate, or that scar substrate. So one of the critical features of whether or not this reentry will set up is actually that conduction velocity I mentioned. If that conduction velocity is fast enough that this wave can circle around and catch its own refractory tail, then it'll terminate, it'll block, just like we mentioned before. However, if the conduction's slow enough, or the refractory period is short enough, or this path is long enough, then you can get the conditions of reentry. So there's a couple things that come into play. How long that path is, how long the refractory period lasts, and the velocity that wave is moving. And those are some of the factors that we really want to start consider as we develop maps of the electrical properties of the heart. Is there electrically non-excitable tissue? How fast is it moving? And all those things that I mentioned. So with no further ado, let's jump into electrical mapping of the heart. So when I say electrical mapping of the heart, how do we create a map of the heart? I don't think the satellites can quite get the resolution they need to map our hearts. So what we do is in clinical cardiology, catheters will be threaded into the inner chambers of the heart in order to record electrical signals. So here you see electrodes on those catheters, and they can move around as they're tracked in 3D space to record electric potentials. What this looks like, again, we'll return to our model, a little model of this reentrant tachycardia. And if we introduce a catheter into that system with a reference electrode, we can move that catheter around sampling electrograms. So here we have an example where we've recorded an electrogram from that point, and this is what the electrogram we get. And we compare it to the reference electrogram measured at that point. So this is our stable reference, won't move. This is our mapping catheter that will move around and explore that tissue. Now, in order to assess what this wavefront is doing, what this reentry is doing, we can look at what's known as the local activation time. This is simply the maximum negative deflection of this electrogram. So the steepest slope of this electrogram is considered as the time when that wave passes the recording electrode. We can measure that property on both signals and then annotate that point with that property. As we do that for multiple points, and we see that those timings go from early to late, we can reconstruct the path of that reentry and tailor treatment strategies to that path of reentry. For example, you might burn at this exit site to prevent that loop from forming. Okay, so this is kind of more our classic electroanatomical mapping. What I'm a little bit more interested in is what we call substrate mapping. And so rather than looking at the timing of these signals, we're actually looking at the morphology of the signals to infer properties of the tissue. So an example of this is what's known as voltage mapping. In a voltage mapping case, it's very similar, where we introduce a catheter into the system, and if we record, in this case, a bipolar electrogram, so the potential difference between these two most distal electrodes on the catheter, we get an electrogram that looks something like this, and we can quantify the bipolar amplitude of that signal. Pretty easy metric. And now we can move the catheter from scar tissue to progressively less scar and into healthy regions. And as we do that, we get an increase in that bipolar amplitude till we're seeing potentials like this. So voltage in this case can be a somewhat indirect marker of the underlying properties of the tissue. And this is something that's easily done. Here's an example that we did in a canine right atrium where we were able to map the voltage surrounding lesions in this right atrium. So you can see low voltages here where there's a lesion here and a lesion here. You see low voltages surrounding it, and areas removed from those lesions, we have higher potentials. So one other type of substrate mapping that I wanted to discuss, and there are multiple kinds, but here's another one that has interested me, is conduction velocity mapping. So this is widely used in experimental settings, but doesn't necessarily have robust clinical application at this time. The way this is performed is you have a heart, and you have a dense electrode array that's placed on that heart. So each of these black circles represents an electrode on that array. We can stimulate from that electrode array and evoke an action potential impulse from that stimulus. So here we have electrical activity spreading off. That's like each color is a time shot of that wavefront moving off this plaque. And there's properties that we can ascribe to this. One of the first things you'll notice is that it's elliptical. This is due to the muscle fibers in the heart. So the heart, like skeletal muscle, has fibers, and this conduction is going to propagate faster along the fiber direction than it will across it. That's something with this type of data we can represent, or we can quantify, so we can get longitudinal conduction velocities or transverse conduction velocities, or we can use that data in conjunction to compute an anisotropy ratio, which is simply the ratio of the longitudinal over the transverse conduction velocity. Okay, so those are the basic mapping strategies that we're going to be considering for our purposes today. Now, I want to move into my specific aims, but we have one more hurdle. Noticeably absent from my discussion is what this thing is at the center of our map. Well, this is a computational model of a cardiac electrophysiological system. Specifically, it's the way that we are able to test out different mapping approaches. So let's briefly cross what I'm going to call our methods mountain. So there's even a substrate on this map. And discuss how these computational models are developed in order to evaluate these mapping systems. So for our simulations, we implement what is known as the bidomain model. The bidomain model is a mathematical formalism that averages the electrical properties of cells over a geometric model of the heart. So it's discretized in a manner like this, where you can get those local electrical properties spread out over a larger domain. From these type of models, we are able to reconstruct the spread of action potentials, or simulate the spread of action potentials with a high degree of fidelity to actual physiological systems through these computational models. Another important feature about the biodomain simulation is that we can calculate what extracellular potentials would be under those conditions of activation. So as you see this wavefront moving through this tissue, it produces extracellular potentials that can be sampled by virtual electrodes that we place into that model. And that's going to be the premise of how we sample electrograms and do this type of substrate mapping for the remainder of these aims. I will mention real quick that we currently use what is known as the Cardiac Arrhythmia Research Package, developed by collaborators Ed Vigmund and Gernot Planck, who've been very kind to help me get started on this and provide us a license of this software. Okay, so here's what a typical model I'm going to use will look like. It's called a slab model, and like I said, we can introduce realistic geometries from the clinical cath lab So this would be like a mapping and ablation catheter that might be used in an electrophysiology study. We can stimulate wavefronts that move through this tissue. And additionally, we can incorporate features such as non-excitable lesions. We can tune conductivities to vary the conduction velocity of these waves that propagate through the medium. And we can change the fiber orientation of this model to affect how those features interact with our substrate maps. Okay. I guess one final point is we immerse this whole system in a bath to simulate the environment in which we're recording electrograms from inside the heart. So with no further ado, let's jump into aim one. In aim one, what I wanted to do is evaluate the performance of voltage mapping to be able to detect non-excitable or diseased features of a cardiac substrate. So to do that, I simply incorporated lesions or scar into this model of various size and also incorporated a representation of a mapping and ablation catheter. I moved that mapping and ablation catheter around to multiple locations to sample electrograms. So what do we expect to see as we sample electrograms from the surface of that model? Well, we might anticipate that if we track our catheter location, whether it's touching scar or whether it's touching myocardium, that we'd see a different bipolar amplitude depending what sort of tissue we're touching. We would predict that in scar tissue, there'd be a lower bipolar amplitude, and in healthy myocardium, there would be a discernibly higher bipolar amplitude on our electrograms. Now, this is maybe a little bit too idealistic. It might end up looking something a little bit more akin to this sort of transition. But that's what we would hopefully see from this data. The trick with voltage mapping is, as I mentioned before, these are bipolar electrograms acquired from the two distal electrodes on this catheter tip, or between two electrodes that are in close or that are in proximity to each other. Now, the problem with this is that because of this positioning, there can be geometric factors that influence the morphology of their electrograms. As an example, if I take this mapping and ablation catheter and rotate it 180 degrees, as we pass impulses through this tissue, you can see the change in morphology that occurs from the one end of the spectrum, it goes like this, to a morphology, this negative morphology, as you rotate it 180 degrees. Additionally, so I'm going to call that the wavefront alignment of the catheter, or the alignment of the catheter with respect to the wave. In addition to that geometric factor, there's also what I'm going to refer to as inclination or inclination angle. And this has to do with the idea that as a catheter is introduced into the heart, it can be difficult to control whether or not this second electrode is in contact with the tissue. We're going to assume that we can get this distal electrode to be touching the tissue, but we could be recording at any of a range of possible orientations. In order to incorporate that into the model, we sampled electrograms at a full range of orientations that can be represented by these spheres, where each sphere represents a position assumed by that second electrode when we keep this distal electrode fixed. So a whole hemisphere of sampled orientations. Okay. So going back more to what we would expect to see. Again, this transition from scar to healthy myocardium. But how can we classify these electrograms to determine whether or not we're successfully discriminating between these tissues? Well, the way that I selected is what's known as a confusion matrix. So in a confusion matrix, it's much like that plot I showed you before, where here we're tracking what type of tissue that catheter is touching, that distal electrode on the catheter is in contact with, whether it's scar or ablation lesion or normal tissue. And here on the y-axis, we have the bipolar amplitude. Now, to classify or discriminate between these two tissue types, we have to set a threshold, a voltage threshold, and we'll call everything under that scar and everything above that healthy tissue. But of course, there could be some noise in that model. Consequently, any electrogram that's acquired from lesion and is below that threshold is what's known as a true positive. And conversely, anything sampled from normal tissue that's above that threshold is a true negative. So and then every electrogram we sample can be classified into one of these four categories. Okay, so we can classify our electrograms, but how do we evaluate that data? We use what's known as a receiver operating characteristic curve, or an ROC curve. And what is done here, simply put, is if you vary this threshold up and down, you get a new point on one of these curves. If you have a very sensitive, a high performance metric, you're going to get a curve that approaches a high true positive ratio and a low false positive ratio. So sorry, I should have said that at the beginning. On the y-axis is the true positive ratio, and x-axis is the false positive ratio that are computed from this confusion matrix. So if your curve approaches this corner, it's a very high performance classification. On the other hand, if it runs diagonal, that means you have almost zero predictive value from your classification scheme. So we applied this to our model for a number of different parameters. For the sake of time, I'm going to present just one of those today. Hopefully is the most interesting. And this would be the dependence of the inclination angle of the catheter on the effect that it has on voltage mapping. So when we look at an ROC curve of the generated based on inclination, well, first let me show you the baseline data. So when we look at all electrograms sampled and classified into those 4 categories, we get an ROC curve that looks like this. So not terrible performance, but there's room for improvement. However, if we remove electrograms that were sampled below 15 degrees of inclination, so if we remove electrograms sampled with both electrodes in contact with the tissue, we see a significant jump in the performance on our ROC curve. And as we remove lower and lower electrograms, you can see that our performance goes up and eventually plateaus. But what this seems to indicate is that if you record electrograms with the catheter oriented perpendicularly to the tissue, you're going to get much better performance in characterizing the different tissue types. Okay, so we thought this was pretty interesting, but we wanted one other way to look at it. And so returning again to our expected results, I've got a plot that I think helps explain what's going on here in a little bit easier terms. So if we plot, again, we're plotting the position of the catheter. This time, though, we're plotting the position of the catheter relative to its distance from the border of the scar tissue. So a point over here will be well within a scarred tissue, and a point over here will be well removed from the scar tissue. Again, the y-axis is bipolar amplitude, and we've colored these plots based on the inclination angle of that catheter recording. So what we see here, if we look at these blue electrograms, these are our low inclination angle electrograms, is that there's a huge amount of variance in the bipolar amplitude of those signals. Regardless, I mean, they do decline in amplitude as we get into scar tissue, but regardless, they're all over the map. Once we start looking at higher inclination angle electrograms, let's say even above 40 degrees, we see this nice tight band that follows what are kind of a priori predicted results. That is to say, we get this transition from low voltage recordings to discernibly higher amplitude bipolar amplitudes. So again, like I said, we thought this interesting, and fortunately, the folks at the Journal of Cardiovascular Electrophysiology felt the same, and they were nice enough to put this figure on the cover of their journal. It was last July or August. I think it was July. But we're not done there. So in terms of future work, we wanted to think about how we could incorporate this inclination angle into acquiring better electroanatomical maps. So what I've done there is I realized that using current clinical systems, it's really difficult to control that inclination angle of the catheter. So what we did is we took, I guess you might say, somewhat existing prototype of a basket catheter. So this is an electrode array, a multi-electrode array, in which there's electrodes placed on splines that form a basket that can be deployed in the heart. Now, instead of sampling bipolar electrograms between electrodes on the splines, the technique we want to try out here is to record bipolar electrograms between a central reference electrode that's surrounded by this basket. So now, if we compute a bipolar electrogram between those electrodes on the spline and the central reference electrode, we can ensure that we always have an optimal inclination angle for our recordings, and hopefully really increase the sensitivity and performance of voltage mapping procedures. Right now, we're in talks with a company to try and prototype this catheter. Okay. Moving on to conduction mapping, or more specifically, conduction velocity mapping. As I mentioned, this is a technique that's used frequently in experimental settings and does a good job of discriminating between healthy and diseased tissues. What we lack is a robust and meaningful way to perform this same type of analysis in the clinical setting. What we might propose is something along these lines. Is you navigate the catheter through the tissue, stimulate the tissue to evoke an action potential impulse, and then characterize that impulse based on its spread through the tissue. Then we can move to other locations, repeat that process, and create a map over this domain that illustrates the local conduction velocity properties. As I mentioned before, conduction velocity is a key parameter in the development of rhythmogenesis and has been shown in experimental settings to be a very sensitive parameter to alterations in the myocardial substrate. Well, how can we get this into the clinic? This type of device isn't going to cut it to be able to characterize that complex activation pattern. Well, let's go back to what's done in the experimental setting. Again, high-density electroarrays that can record electrograms from over a large spatial domain in order to characterize the propagation of the wave through the myocardium. And it just so happened there was a colleague named Dr. Anders Peter Larsen up at CVRTI that was doing just this type of mapping. So he'd stimulate the tissue, evoke an action potential, and use that to characterize the conduction velocity, longitudinal, transverse, and anisotropy ratio like we discussed before. To do this, he would look at the electrogram produced from each electrode and assess the activation time on that signal. So pretty basic. The only problem is that these unipolar electrograms are notoriously sensitive to noise and become fractionated, making it difficult to assign a local activation time. Well, there's a technique called Laplacian electrograms, where instead of taking the temporal derivative of this signal to find the steepest slope, we actually take the second spatial derivative over this plaque. Now, that can be done with a kernel that's essentially a linear combination of those plaques to produce what we call the Laplacian electrogram. It looks something like this. Now, the Laplacian electrogram has some nice properties in that it's less sensitive to noise, and identification of activation times is easy because the activation time is simply the zero crossing where the signal goes from positive to negative. So up till here, this all makes perfect sense and it is a good way for looking at the data. But at this point, there's kind of a stepping off where Anders did something that was at first kind of inexplicable, especially given the paradigm of looking at each of these individual electrodes to kind of figure out what's going on locally. So what he did is he took the average, or the mean, of all these Laplacian electrograms. So summed them up, divided by n, to find this mean Laplacian electrogram. And what he found is across multiple experiments and through a variety of different settings, that there was this characteristic pattern to the signal where you get your stimulation artifact, and then you get these two peaks. And as he induced changes in the conduction velocity, in this case he introduced flecainide, a sodium channel blocker that's known to slow conduction velocity, he saw that these timings, the timing of these peaks correlated with the conduction velocity. So transverse, or longitudinal and transverse conduction velocity respectively. So this is interesting. There's this nice correlation between these features, but we don't really understand what's going on here. Timings are not velocities. There's no distance aspect. You need a distance over time to get a velocity. And okay, so if these have something to do with the velocity, where's the distance side of this equation? So to get a better understanding of what was going on, Anders approached Rob and I and a few others and asked if we could perform some simulations to see if we could reproduce this and might be able to figure out what was producing this signal. So what we see here is we created another simple model where we stimulated from the center. You can see it's anisotropic with fiber orientation diagonally in that direction. And we included a representation of this plaque electrode array in our system. And lo and behold, we were able to sample this two-peaked mean Laplacian electrogram. And furthermore, these peaks, again, corresponded to the conduction velocity that we measured in our model. So we were able to reproduce this effect. The only problem we had is that when with this model and the easy ability to permute the system or to perturb the system, we rotated that plaque with respect to the fiber orientation and found that that characteristic signal quickly decayed. At this point, we realized that maybe just correlation wasn't sufficient. We really needed a better understanding of how this wave was producing this type of signal, if it was to be of any use at all. So in conjunction with Dr. Peter Johnston, who was visiting here at the time, we were able to do an analysis of this calculation of the mean Laplacian electrogram. So as I mentioned before, here is the kernel that was used to calculate the Laplacian electrogram. And that's calculated by applying that kernel over this electrode array. But if we sum those potentials, so we sum each one, you can see that that linear combination has an unintended effect where these central electrodes end up canceling out. They go to 0. So what are we left with? Well, we're left with two rings of electrodes, one negatively weighted, one positively weighted. We're calculating bipolar electrograms between these two rings of electrodes. That's something we know about. So in order to get rid of that rotational anisotropy, that rotational asymmetry, we simply converted this plaque electrode array into a simpler design of two ring electrodes from which we paste at the center. Once again, we incorporated that into our model, and we're able to, once again, get that characteristic two-peaked signal. Okay, and I should mention that as we rotated this array, it, not to say it was perfect, but it was much less sensitive to the rotational aspects we saw before, dependent on the sampling of these electrodes. The other thing we can do from this simulation is a direct comparison of the conduction events relative to the features of this electrogram. So if we pause that simulation, when we see these features, so we see this first peak at 9 milliseconds, what we find is at 9 milliseconds, the longitudinal aspect of this wavefront is passing over the outer ring. And at this second peak, the transverse aspect of this wavefront is passing over the inner ring. Finally, this zero crossing corresponds to the propagation of this transverse aspect over the second ring. So now we have a direct comparison of what conduction events are producing these signals. And why is this important? Because of the distance. Now we know where that signal is and when it's there, and we can get from this signal, from the signal, we can now directly compute conduction velocities. Real quick, the second peak, although it has a clear correlate in the propagation, we found that if we tune the model so it becomes more isotropic, this second peak begins to overlap with the first peak and can be difficult to detect. Because of that, and because both of the first peak and the zero crossing correspond to this outer ring, we decided to proceed further to see how these rings, how those features were able to predict conduction velocity compared to standard metrics. So when we compared, from those, here we go. Just thought you might want to see it again. So if we calculate conduction velocity using standard activation time-based approaches and compare that to conduction velocities calculated from the mean Laplacian electrogram, you can see that we get a high correlation. This is the longitudinal or first peak component, and this is the transverse component derived from the zero crossing. And there's an extremely high correlation between the two. Why is this interesting? Well, you're left with two options at this point. You can look at that plaque array, and you can try to find activation times from 64 or more noisy channels, or you can do this whole array processing and identify two features from one signal to get the same data. So this mean Laplacian electrogram provides maybe a new insight for how we can look at the sampling of electrograms from these multi-electrode arrays. So rather than be concerning, okay, what's happening here, if we can get a more holistic view using current clinical systems, I mean, there's all sorts of systems out there right now that can be deployed in the cath lab. There's the potential to simplify the characterization of the cardiac substrate. So with that in mind, final point on this. Unfortunately, this dual ring electrode array does not currently exist in the clinical setting. Great if it did, and we'd love to work more with it. But for the time being, we want to focus our efforts on arrays and devices that are currently in existence to try and port some of these ideas into the clinical setting now. So with that, we'll move on to the third aim. And so the third aim, we're looking at what I call activation pattern mapping to reconstruct the conduction velocities. So one device that rose to the top in our thinking is known as a lasso catheter. And this lasso catheter has some really nice properties. First and foremost is the fact that this lasso catheter is used in virtually every AFib ablation case. Deployment is ubiquitous. Additionally, there's capabilities to track this lasso array so that we can know the position of every electrode in the heart. And as you've seen before with conduction velocities, positions and distances and timings are very important. So what our plan here was to stimulate with this electrode array and then record from all the other electrodes on that array. So we stimulate there, and then we stimulate around that array and produce multiple wavefronts moving from the tissue. For those of you more familiar with imaging, this will start to trigger thoughts about computed tomography or approaches in which you stimulate or you pass a wave through a medium from multiple directions to reconstruct the relevant features of that medium. Well, once again, we turn to a simple slab model to test this type of stimulation pattern. So we stimulate between electrodes and produce activation waves that move through the tissue. As interesting as that is, let's see what we actually can produce when we use that type of stimulation pattern. So here we have a body surface electrode. And I'm cheating here. I'm actually showing you animal data rather than simulation data, but hopefully that's even a little bit more convincing. So here we see from this body surface electrogram is a pacing artifact. So we've stimulated the tissue, and then we see the ventricular response. And as late as it is, we know that we're pacing in the atria. If we look a little bit closer at these electrograms, here again we see the pacing artifact. Here's the electrode that we paced from. And if we get away a little bit from that electrode array, we start to see these signals in the electrograms that represent that wave front passing by those electrodes. Subsequently, if we analyze those electrograms for activation times, we now have timings and we have distances. We can compute conduction velocities. Additionally, because we've stimulated the tissue through the full range of orientations, we can have the ability to reconstruct the longitudinal and transverse aspects of the conduction velocity locally. So in this case, we started out with an isotropic model. So what that means, here we have a nearly circular activation pattern. So conduction is the same in all directions. We should be able to reconstruct that pretty easily. And sure enough, when we reconstruct those conduction velocities, depicted here as vectors pointing away from the stimulus site, so this vector was stimulated here, the coloration and size of those vectors is indicative of the velocity of the wavefront as it moved to that point. So you can see that at each point we can get a full range of conduction velocities. Now what happens if we try a more anisotropic, try this in a more anisotropic model? So you can see we get a more elliptical propagation pattern, and if we evaluate those conduction velocities, you can see we can recover the anisotropy of the system. And clearly, these arrows indicate the fiber orientation. And here we see that the conduction velocity is much faster along those fibers than it is transverse, in the transverse direction. Okay, so with some signal processing that was necessary to produce these conduction velocity vectors, we were able to present these results last year at Computing and Cardiology in Boston. So what do we learn from this? Well, we want to do this clinical propagation or conduction velocity mapping. The advantage of using a system like this is that it's immediately clinically feasible. We have the devices we need. It's simply the processing that needs to be brought up to speed. Additionally, through this type of stimulation pattern, we can evoke a response from multiple different orientations and characterize the tissue response to multiple activation patterns. And we believe by recovering these conduction velocities, we are in fact measuring a direct measure of the tissue's pro-arrhythmic potential. So in terms of future work for this project, we're already moving forward as quickly as we can. And we've been able to perform this type of mapping in the clinical setting in patients. So here we have that type of sampling. In this case, we projected all those vectors that were at the electrodes to the center of this array. And you can see in this patient, this is the posterior wall of the left atrium. There's a strong longitudinal and transverse component. With some simple signal processing, specifically a singular value decomposition, we can reconstruct the principal components of this conduction that suggest that the longitudinal conduction velocity moves from an inferior to superior direction, and that the transverse conduction moves between the pulmonary veins. Now, I'm going to say the reason I put this in future work is I want to speculate a little bit on this. When we look at histology from patients, so this is the actual left atrium of a human, we see that on this posterior wall, there's this inferior to superior orientation of the muscle fibers, which over a number of patients, we seem to be able to reproduce. So there's definitely some work to be done in the signal processing, but at this stage, we already seem to be able to recover such features as the fiber orientation and presumably the anisotropy ratio of the conduction. Okay, one last final thought on that point. So, and I briefly mentioned this before. With this multiple activation pattern approach, we really think there's a potential to do this tomographic substrate reconstruction. Just as in CT, where X-rays passing through the medium reveal the interesting substrate of the body, be it bones or liver or obviously the heart, with these multiple activation patterns, we believe that we'll be able to reconstruct the relevant substrate in hearts of patients with atrial fibrillation. Okay, so finally, let's talk about how these maps compare and what kind of information they contain. So in the voltage mapping study, we were able to show that voltage mapping does have a high ability to discern between scarred tissue and healthy myocardium, and that if we can control the inclination angle of our bipolar amplitude recordings, that we can get a much higher performance in this classification. Additionally, we showed with the mean Laplacian electrogram project that a holistic approach to analysis of electrograms coming from our arrays can simplify the process of characterizing the substrate and open up new pathways that don't require huge arrays of electrodes, but they can actually be simplified down to more clinically feasible type devices. And finally, with that type of strategy in mind, we were able to translate this approach onto a clinical device and show its feasibility for application in the clinical setting. With this type of mapping, I can't say at this point that any of these maps are going to revolutionize atrial fibrillation, but I do think we're on the right track here. I think as we get a better understanding of the proarrhythmic substrate in patients with atrial fibrillation, that we'll better be able to design treatment strategies for patients to move from the atrial fibrillation side of the AF canyon over to the sinus rhythm side. And as with any crossing, whether it be from atrial fibrillation to sinus rhythm, or from the north rim to the south rim, grad school is another major crossing for someone. And it's also one of those crossings that's very difficult to do alone. And I'm very appreciative of those who have had a part in helping me make this crossing. There's obviously too many to mention here. I'll give special thanks to my committee, who have provided me opportunities on, as Rob mentioned, multiple different projects, and have been great mentors and teachers for me. And then last but not least, I would also like to thank my family that has been there with me the whole way. And to end, as Rob mentioned, here are some publications, briefly a book chapter and some conference proceedings, and then journal articles that I've been involved with as an author. And I would be very happy to take any questions you might have.
All right, so as I said before, this is the open question period, and Josh has some microphones over there, which I'll ask you to use as you ask questions so that Frank can participate from Germany and hear our questions and obviously Josh's answers, and also pose his own questions. So, Frank, hearing?
Those are really nice results, but I noticed that most of your sensors were two-dimensional. Do you think things might change with 3D approaches?
To be honest, in the latter two aims, they were more two-dimensional. In the original aim, it was a three-dimensional structure. And definitely, there can be influences when you're sampling electric fields if your catheter has a geometry as well. It's really convenient to assume that your catheter is, void of any dimensionality or geometry, but it does have an effect. And we also tend to like to assume that our electrodes are perfect conductors, and they're able to sample these computed electric fields just as an electrode in the clinic might. But that definitely can have an influence. I did spend a fair amount of time testing out different sampling schemes, and what I found when I create a three-dimensional electrode, so there's models in which you can actually incorporate a real representation of an electrode into your mesh, as it were. And when you sample on those, you probably get the best reproduction of an electrogram. In my case, I found that if I did an average over the surface of that electrogram, that I was able to really closely reproduce the same type of potentials, which made it a lot easier to calculate our electrograms, especially the thousands of electrograms that were necessary for these studies.
That was a really great presentation. Thanks for that. I learned a lot. So I guess this is a question, for you and Nasir. When you were doing your simulations, you were sampling for these hundreds or thousands of points in your substrate. What's the obviously clinical implications in terms of how much time can Nasir take to sample the substrate, and is there an optimization problem here of what might be the optimal number of samples you need to reconstruct the electrograms sufficiently?
Clearly, a key aspect of maps is what resolution are you working with. If I try and look at the Grand Canyon as a map of 2 pixels, I could be seriously misled. And I think the same, and I'll allow Nasir or Ravi to contribute to that discussion as well if they feel like it. But the resolution here is going to matter as well. Interestingly, about the same time I published that first paper, another group published a study in which they provided a mathematical basis by which sampling electrograms at a higher inclination angle, so perpendicular to the substrate, produced a higher resolution maps of those substrate features. Now, what sort of time do we have to sample all those electrograms? And part of my answer for that would be, and part of where I see electrophysiology moving, is the use of multi-electrode arrays to sample those electrograms. So rather than doing a point-by-point interrogation of your system, you have an electrode array that can sample many electrograms at once. And this has some pros and cons. One, you have less supervision of what type of electrograms you're incorporating into your model. Or if you're doing it one by one, you can make sure, okay, yeah, that looks good, that looks good. But as you get multiple electrograms, it increases the speed of your mapping, but it does have that limitation. But that's why I proposed that basket electrode, where we have a basket electrode where we can sample multiple electrograms at once, but by constraining the sampling orientation so that we get those high inclination angle recordings, optimistically at least, we're getting the best possible electrograms as quickly as possible.
Okay, thanks.
Oh, it's a great presentation. My question is with the mean Laplacian signal with the 2 peaks. So I was just curious how you think those curves might change if you have one, because you have 2 transverse and 2 longitudinal directions. What if one of them's slower than another? How will that shift your curves, or what will that do? Because both of yours hit the same time. Or what if one of those sides happens to block or not? If you can actually detect these things.
That is, that definitely falls into the category of future work. Yeah, assuming this technique might work great in healthy myocardium. And okay, you can map all this healthy tissue, but what does it serve? What you arguably want to map is you want to find these locations of block. You want to find the disruption of conduction in the heart. And so if you don't get this nice symmetrical spread of activation, then it is going to disrupt that signal. And that goes beyond what we've currently tested, how that impacts the morphology of our signal, and whether it really even makes that a reasonable approach for trying to map real clinically diseased substrates. Ed?
To the passing thing, my question is, it seems to me that the orientation dependence of the passing operator is more like a numerical artifact because of the...
Absolutely.
Right. So is there a numerical solution to that instead of having to redesign, a new electrode arrangement?
I do imagine you could change your sampling to account for that effect. We haven't really considered that in depth. But yeah, I think actually that would be a good approach to take, rather than whoever uses a plaque electrode having to, redesign that plaque electrode. Why not change? I mean, the mathematics are there. It's just going to be a different sampling scheme. I'm confident you could. Can't tell you exactly how to do it at this moment, but I'm confident you could.
Final question.
Here comes the uncertainty question.
No, this is I don't know, maybe this is easier, maybe it's hard. So assuming the deliberations go positively from your committee, what's your plan after this?
Oh, thank you. About midway through, I looked over and I saw Chris and I said, I'm going to get an uncertainty question. And from then I'm like, okay, is it going to be on this slide or this slide? Like, what is he going to ask?
I thought of uncertainty.
Your reputation has preceded you, but I guess I'm getting off easy. So after this, I am starting a postdoc with Derek Dosdall. So I'm actually doing 2 postdocs, one in collaboration with Dr. Ranjan and Natalia Trainova from Johns Hopkins. We're doing some clinical studies looking at the use of some of these models, more patient-specific, anatomically appropriate models for characters or for prediction of, without going into detail, prediction of treatment strategies. And then additionally, I'll be working with Derek Dosdall up at SMBB, working on some defibrillation, in a way defibrillation studies, VF studies. I don't know if that information is necessarily... Thank you.
Okay, are there other questions from the audience? Otherwise, we will have to ask you to leave. All right, well, if not, then thank you all for coming. Let's thank Josh one more time.